Hi there. In this video we are going to discuss certain aspects of cell biochemistry. Cell biochemistry, and it's an important part of first year MBBS, for first year MBBS. Okay. So in this lecture we will have a discussion about the cell membranes. So when we come about the cell membranes, we know that cell membranes have glycolipids, they have phospholipids, they have sphingolipids, they have sterols, and so on. So different kinds of lipid found in the cell membranes are amphipathic in nature. They are amphipathic in nature. What does it mean? It means that they have both polar and non-polar regions. They have both polar and non-polar regions. When we come towards a typical phospholipid, we know that phospholipid have one head region and a tail region. This head region is polar or hydrophilic. However, this tail region is non-polar. It's non-polar or hydrophobic. It means that this polar region will interact with water. It interacts with H2O. However, this region flies away. It runs away from water. So membrane lipids are phospholipids found in membrane are amphipathic compounds having both polar and non-polar regions. And this tail is straight. It's straight when membrane lipids are saturated. When membrane lipids are saturated, it means that when membrane lipids don't have any double bond, this tail is straight. However, when they have a double bond, it gets tilted. A nick is introduced here. Nick or a tilt is introduced here when they become unsaturated or when there is some double bond. Okay. So these membrane phospholipids, on one side they are amphipathic, and on other side there is a interaction with water by polar region, and non-polar region flies away from the water. And moreover, the saturated ones have straight tails, while unsaturated have non-straight or tilted tails. Okay. Another thing we should be understanding here in the context of amphipathic compound is role of detergents. So what are detergents? Detergents are similarly amphipathic compound having both polar and non-polar regions. What happens? Supposedly this is a piece of cloth here, and here is some mark of a hydrophobic compound. So what happens? This is supposedly a detergent molecule. This detergent molecule has polar and non-polar regions. Its polar region here interacts with water H2O, while non-polar regions interacts with this mark here, and this interaction lead to the dissolution of this mark, which may be some hydrophobic compound or protein and so on. So this is the action of detergents, in which non-polar region of detergent interacts with the non-polar mark here on cloths, and polar region interacts with water, and this interaction lead to the removal of this mark from the cloths. And similarly, detergents are used to dissolve membrane proteins as well. They are used to dissolve membrane proteins as well in the cells or from the cells. So this was a basic overview of the membrane lipids. Now we should know how these membrane lipids respond to the aqueous environment. Supposedly these are the multiple lipid molecules, phospholipid molecules here. Okay, and they are introduced to water. We pour water H2O here. Okay, so what will happen? The polar region they will rearrange themselves in such a way that their polar region interacts with water and non-polar region flies away from the water. So what will happen? They will attain a shape like this. Okay, they will arrange themselves like this. Okay, so this is a rearrangement, and here will be water molecules. Okay, so what has happened here in this configuration? the polar region or polar heads are interacting with the water while non-polar tails flew away from the water. So this configuration is missile formation. So lipids or phospholipids, when they are exposed to water, they form micelles. Similarly, detergents also form micelles in the water, in which their polar region interacts with the water and non-polar region flies away from the water. Another configuration which can be formed here, when lipids are exposed to the water is this configuration. This configuration, lipids can also form this configuration. And what is this configuration? This configuration is a lipid. It is a lipid bilayer. So in this configuration, here is water, here will be water molecules H2O, and here will be water molecules H2O. So in this configuration, what has happened? Polar region is interacting with water while non-polar tails are shielded away from the water. So this is lipid bilayer, which also formed when lipids are exposed to water. So this is the configuration of phospholipids found in the cellular membranes. In the cellular membranes, lipids attain this configuration because there is water in extracellular environment as well, and as well as intercellular environment. A cytoplasm also contains water. So lipids arrange themselves in this way in the cells or the cellular membranes, in which they form a lipid bilayer, in which their polar heads interact with water and non-polar regions are shielded away from the water in this bilayer structure. So in this bilayer is permeable to the lipid soluble substances. It is permeable to lipid soluble substances. For example, oxygen, carbon dioxide, nitrogen can cross this lipid bilayer easily. Moreover, the lipid soluble substances, other lipid soluble substances can also cross it easily. However, the ions, for example, charged particle Na positive, K positive, they cannot cross it easily. So in order to cross this lipid bilayer, they need certain carrier proteins. And these carrier protein can be in the form of carrier protein, or they can be in the form of a protein channel. So these ions cannot cross it or permeate this membrane. So this membrane plays its role of semi-permeable. It plays semi-permeable role, and these positively charged particles and certain other molecules, they are transported across this membrane with the help of certain channels or carrier proteins. So next important component of cellular membrane here is proteins. First component we discussed are the lipids, and second component comes here, which are proteins. Okay. So what are these proteins? Let's have an overview of these proteins here. Okay. So as lipids were amphipathic compounds, membrane proteins also show a amphipathic behavior. So this is a membrane here supposedly. Okay, I am drawing a rough membrane here. So membrane proteins also have both polar as well as non-polar regions. They both have polar as well as non-polar regions. Membrane proteins cross this lipid bilayer. When they are crossing this lipid bilayer, they are in alpha helical configuration. Alpha helical configuration, we know that protein have primary structure, secondary structure, tertiary structure, and so on. So this alpha helical configuration imparts non-polar nature to the protein. So in alpha helical configuration, proteins are non-polar. So part of protein which crosses this membrane is in alpha helical configuration. For example, supposedly this is protein crossing membrane. This region will be in alpha helical configuration, while the ends of protein, they can be they are in higher structures, and they are usually polar, as they are interacting with the aqueous environment or polar environment. So they are polar. So this protein also showing a amphipathic nature. So in order to cross this lipid bilayer, polypeptide chain of 20 amino acids are enough. 20 amino acids are enough to cross this membrane, and usually protein which cross this membrane, their region which cross this membrane is usually made up of amino acid which also have a hydrophobic nature. And moreover, the proteins can also interact with lipid bilayer outside. Okay, here they can interact, which this phenomena is called lipidation protein lipidation, in which proteins are added up to the lipids outside. Okay, so another aspect here is cellular membranes are dynamic structures. They are dynamic structures. It means that their configuration does not remain constant. These lipids, they also show turnover rates and replaced. They also show turnover rates and replaced by new ones. Protein also show turnover rates and replaced by new one. They also show lateral mobility. What is this lateral mobility or lateral? You can say it lateral diffusion, in which these lipids move here on the lateral side. They continue to move, and in turn previous new lipids take their place. Similarly, these proteins also continue to move or show lateral diffusion and replaced by new proteins, so on. So this is another important phenomena in the membrane, the structural membrane. So membranes are basically dynamic structures which also show the phenomena of lateral diffusion, and newer proteins take the place of older proteins. Okay, so another thing we should be knowing here about the membrane proteins form asymmetric structures. Membrane proteins are asymmetric structures. Asymmetric structure. What does it mean? It means that internal and outer configuration of protein of the membrane protein are different. For example, we know that in respect of membrane protein have G proteins which interact with G protein coupled receptor. So these G proteins are not found on the outer aspect. Similarly, outer aspect will have certain proteins which are not found on the inner aspect. So membrane proteins are asymmetric structures. Their structure on the inside and outside does not match. For example, let us discuss the choline containing phospholipids. For example, phosphatidyl choline and sphingomyelin. They are present on the outer aspect. Phosphatidyl choline and sphingomyelin present on the outer aspect of membrane. However, the amino phospholipid which contain amino acid, for example, phosphatidyl serine and phosphatidyl ethanolamine, they are present on the inner side of cellular membrane. You just need to remember the names of these lipids here. Phosphatidyl choline and sphingomyelin are located on the outer aspect, while phosphatidyl serine and phosphatidyl ethanolamine are located on the inner aspect of cellular membranes. So this imparts asymmetry to the cellular membrane. Okay, moreover, the glycolipids and glycoproteins are also located on the outer aspect of the cellular membrane. Glycolipids, they are also present glycolipids and glycoproteins which are on the outer aspect, and they also impart asymmetry to the cellular membranes. So cellular membranes are asymmetric structures. Next, we discuss about the types of proteins found in the cellular membrane. So proteins found in the cellular membrane, they are generally divided into two categories. One are integral proteins, and other one are peripheral proteins. So supposedly this is the cellular membrane here. Okay, I am drawing it just roughly here. So this is supposedly a cellular membrane. So what are integral proteins? Integral proteins are those which cross the membrane lipid bilayer. However, peripheral proteins don't cross. Instead, they are attached peripherally. For example, if a protein crosses this lipid bilayer in this way, this is a integral protein. However, the peripheral proteins don't cross. Instead, they are attached at the periphery of membrane. These are peripheral proteins and these are integral proteins. Okay, so these integral proteins, as they are crossing the membrane, they can form channels. Okay, they can be in this form, for example, in this form, and they can allow the certain ions, for example, sodium ion, to diffuse into the cell and so on. So they can form channels. Okay, this channel can be ligand gated, they can be voltage gated, okay, or they can form carrier proteins. They can form carrier proteins. Okay, so carrier proteins are another configuration of protein. For example, supposedly this is a carrier protein here. This carrier protein exists in two configuration, ping state and pong state. So what are these states? For example, this is a carrier protein. Here binds an ion. It can be sodium, for example, and when it binds to sodium, it is a ping state. And what happens? It induces a conformational change in this protein. What happens? It changes its shape, and this sodium shifts to the inside. So this is now pong state. So it flips, carrier protein flips between ping and pong states and transfer the material across the cellular membrane. So proteins in the cell membrane are of two categories: integral protein and peripheral protein. Integral protein cross the cell membrane, while peripheral proteins are attached at the periphery. And these integral proteins can be channel proteins. Okay, channel protein exist in the form of channel and allow the exchange of materials, or they can be carrier proteins. Carrier proteins exist in two states, ping state and pong state. They flip back and forth between these states and transfer the materials across the cellular membranes. Okay, so an example of proteins here is RBC membrane protein. RBCs have a band 3 integral protein. For example, supposedly this is a band 3 integral protein, and on the periphery they have attached other proteins, and these proteins impart the integrity to the RBC membrane. For example, an chirin protein is attached to this integral protein, and it also interacts with spectrin, which is the cytoskeletal element of RBC, and maintains the membrane. It is just for the sake of our knowledge here. So what is the source content of this lecture? This lecture is from Harper's chemistry. My book is too much here, you can say colorful. So this is from the Harper chapter 40, which is membrane structure and function, and it is for first year MBBS. So you should be knowing the membrane lipids are amphiphilic compounds. And next here is the how detergents work, and membrane lipids form bilayers, and then membrane protein associated with lipid bilayer, and membranes are dynamic structure, and then membranes form asymmetric structure, and membrane contain integral and peripheral proteins. So it was all about this lecture. Thank you.